Hi everyone, my name is Joke de Vocht, and today I would like to talk more about the following poster. The majority of ALS patients have a form of the condition that is described as sporadic ALS, while 10% of patients have a familial form of ALS. In about 60 to 80% of familial ALS and 10% of sporadic ALS, gene mutations of large effect are present. The most commonly identified genetic mutations found in a European and Caucasian population are mutations in the C9orf72 gene and in the SOD1 gene. Little research, however, has been done into the cerebral glucose metabolic correlates of monogenic forms of ALS. So in this study, we focused on the glucose metabolic pattern associated with different genotypes of ALS in a large cohort of ALS patients using FDG PET. In our cohort of ALS patients in the Neuromuscular Center in Leuven, we identified 18 SOD1 mutation carriers and 39 C9orf72 mutation carriers that also underwent an FDG PET scan during the diagnostic stage. These were compared to an age, gender, and site of onset matched sporadic ALS group, as well as a group of healthy controls. The cohort of 18 SOD1 mutation carriers that we included in the study consisted of multiple variants of SOD1 gene mutations, of which the majority carried the D90A or G93C variant. Scans were obtained during the diagnostic workup of patients and were processed using SPM implemented in MATLAB. In comparison to healthy controls, we found widespread frontotemporal hypometabolism in all ALS groups, especially in the C9orf72 mutation carriers, as you can see on this slide. In the picture on the left, we compared sporadic ALS to C9orf72 ALS and found significant clusters of relative hypometabolism in C9orf72 ALS in the perirelandic region and thalamus. In the picture on the right, we compared SOD1 ALS to C9orf72 ALS and found significant clusters of relative hypometabolism in the perirelandic region and the cingulate cortex. We also found two significant clusters of relative hypermetabolism in the cerebellum brainstem in C9orf72 ALS in comparison to both SOD1 ALS and sporadic ALS. We found no significant differences between SOD1 ALS and sporadic ALS. So what can we conclude from these findings? First of all, we observed widespread frontotemporal involvement in all ALS groups in comparison to healthy controls, and the pattern that we observed in SOD1 mutation carriers was very similar to the pattern observed in sporadic ALS. We also found that FDG PET underscores the involvement of not only the thalamus, but also the perirelandic region and the cerebellum and brainstem in C9orf72 mediated ALS. Lastly, potentially the absence of significant differences between SOD1 ALS and sporadic ALS may be driven by the composition of our cohort, as different variants are associated with different phenotypes, so perhaps also a different pattern of cerebral glucose metabolism. Therefore, multi-center studies are needed to study different SOD1 genetic variants. Thank you for listening.